people who've joined at this point. So thank you all for your attention and coming to learn more about the non-addictive analgesic therapeutic development program here, both small molecules and biologics to treat pain, and the clinical trial is optional. This is a UG3-UH3 mechanism. I am the program director for this program and responsible for small molecules. Anne-Marie Brome handles the biologics for the program, and Mike Oshinsky is our science officer in charge of the pain and gives us the overall direction of the pain portfolio. So NIH is helping to end addiction long-term HEAL Initiative, has pain research priorities, and enhanced pain management is a key one of those. And the underpinnings of that is we want to understand biological underpinnings of chronic pain, accelerate the discovery and preclinical development of non-addictive, and that's really important, non-addictive pain treatments, advance new and non-addictive pain treatments through the clinical pipeline to hopefully inform best practices for effective pain management while minimizing the risk of addiction. The scientific challenges in analgesic development are many. There's specific challenges like complexity of biology and pharmacology, the historical lack of validated targets in this area, the lack of translatable pain measures, the lack of predictable preclinical models, rigorous proof of concept studies have been problematic. And these last three are the ones I think this program is most addressing in terms of this particular RFA, is having robust therapeutic tools at early indication of safety and efficacy through target engagement biomarkers, and hopefully address the lack of resources and expertise that is typically available at academic centers. The program itself is a much broader program and hopes to identify all those things that I just identified through a number of grant mechanisms, contract mechanisms, and intramural resources that are being established to be part of the whole HEAL Analgesic development program. And although I'm only focusing on the optimization stage, there is in the works planning going on for the early stage planning as well as initial therapeutics development, and I'll just briefly touch on that in just a moment. But we're really focusing on deliverables, focusing on the scientific challenges, minimizing the overall program complexity by trying to make these dovetail quite nicely together, and with the end goal of incentivizing the industry to reinvest in this field. So with the realignment of the analgesic development program, we have initial therapeutics development and the optimization stage therapeutics development. The optimization stage is where I'm going to be talking about the RFA we're talking about today. But know that, in fact, the initial therapeutics development is not being forgotten, and we're actively working on funding opportunity announcements that will be coming out in the near future. I just can't give you the date yet. And the goal of those initial therapeutic developments projects are to identify mechanisms, assays, models, biomarkers, proof of efficacy concept, with the goal of identifying hits and getting you to the point where you can enter the optimization stage. The optimization stage is all about taking those hits and optimizing those to leads and eventually clinical candidates, as well as giving you the target engagement biomarkers, hopefully, to get you that early read in the clinic. Our goal, all of these projects, the goal is to get to an IND and first-in-human trials, and hopefully get you the requirements needed to enter into something like EpicNet at the other side. So the RFA we're talking about today is a non-addictive analgesic therapeutics development, small molecules for pain, small molecules and biologics to treat pain, with the overall goal of accelerating the development of novel, non-opioid, non-addictive analgesics, and that's really important, the non-opioid. I'll talk about what's not responsive in just a second. We have 5-year benchmarks. We hope to have at least 5 promising projects at the end of 5 years with, ready for preclinical optimization, and advance at least 3 of those through IND in human safety by the end of the 5-year benchmark, getting those ready for EpicNet or equivalent phase 2 trials. So RFA NS 21-010 is the RFA. It's a UG3-UH3 It's important to notice there is no SBIR version, but small businesses are eligible to apply under the UG3 UH3. It is at the optimization stage here, and it's a milestone-driven cooperative agreement, which means you'll work closely with NIH staff on this project. We hope to identify and conduct the necessary studies to meet the requirements for initiating optimization or IND enabling studies, develop compounds through lead optimization to develop the clinical candidate compounds, support the IND enabling studies, and enter phase 1. And we're going to do this through working closely with you, but also providing access to experts, subject matter experts, scientific consultants that NIH has contracted, as well as access to CROs, as well as intramural and extramural resources can be available too. So things like the PSPP program, the NCATS programs are all potentially available to you to make into this cooperative agreement team that we're talking about. The end goal, as always, identify clinical candidate, identify clinical candidate. I can't say it enough, that is the goal. So your program, your grant application must show me that we're getting all the way through identifying the clinical candidate, running the IND enabling studies, filing the IND, and running a clinical trial either through your own grants or through our contracts, but our goal is to get to that phase 1 clinical trial. Additionally, and this is a real important addition, is we hope to identify a companion biomarker in here for target engagement. And that needs to be a component of the grant application, how you're going to address at least figuring out the feasibility of that and hopefully actually developing it. We want you to seek partnerships and we want you to be ready for phase 2. So you need to plan ahead and think about how you're going to get there. This is just an example, provide resources not readily available to academia. This is sort of what we have in the small molecule space. It's established because we have established programs in small molecules already with contract resources for things like medicinal chemistry, which has with AMRI, but supports not only the synthetic chemistry, but also in vitro ADME, computational chemistry. We have a relational database system through CDE, which is our data management, and it allows us in the optimization phase to take the biological data generated in your labs along with the chemical data and put them together in a relational database, which is very useful down the road. We have PK tox contracts that can handle both small molecules and biologics, as well as drug manufacturing formulations for small molecules, and we're working on trying to figure out how to establish similar things in the biologic space. And clinical trial presently available through our phase 1 clinical contract that we have here at NINDS through a couple different vendors available. What's really important and can't lose sight of is all the disease biology that has to come from your side and has to come from the grant. It needs to be included in your grant application. We don't have that ability to give you that. And so you'll need to make sure that's part of your grant team that you put together. Either through your principal investigators or your co-investigators. But we'll put together a team, and this team will be the cooperative agreement team, lead development team, lead discovery team. It'll include the principal investigator from your side, as well as our consultants, and it includes NIH staff on these teams. And we work very closely to move your project forward. But we do so while trying to protect your intellectual property through having pre-established IP agreements with our consultants, as well as having clauses in our contracts for the manufacturing and medchem where IP is being developed that assigns the IP back to your institution. So overall, we plan to launch up to 17 projects over the next 3 years. We're thinking about 5 projects this first year, and then it will be and we do expect to have attrition in this program. So we expect to see about 5 of those projects advance to the UH3 phase, and that's going to be based on meeting your milestones, having the best set of data, having the biomarker available. All those things will be important aspects for figuring out which projects move forward. Our next receipt date is March 24th, so it's not that far away, but we have continuing receipt dates through June 22nd. Or sorry, June 2022, sorry. So there is a focus on biomarkers, and we want to have a target engagement and pharmacodynamic biomarker here in this program. And so the idea is to develop that, and that'll help us interpret the IND toxicology as well, whoops, as I'm really trying to figure out how to minimize something on my screen here. This just won't go away. I apologize if you're seeing it. There we go. And help us determine the project the human phase 1 dosing. So I've gone over all this, but what do you really need to know, from entry criteria point of view, is you need to have a rigorous biological rationale for the intended approach. You need to have a promising small molecule or biologic starting point for optimization. So you can't be running a screen and saying, I'm going to identify that. The screen has to have already been run, and you need to have the starting points. And you need to have, scientifically sound assays in place to do the optimization if required, to get to and to test the agent so that we can get to a development candidate in a timely fashion down the road. There are 9 participating institutes and centers presently signed on to this FOA. There are 2 that you may notice being missing, NCI and aging. That does not mean if you had a chemotherapy-induced pain project, it's not eligible to apply. It is. It falls under the HEAL mission. It just means in those cases, Michael will be the science officer rather than somebody from NCI or DJ. So we'll have a couple of different options there. And we do expect some of these other institutes may sign on in the coming months, but presently that's where we're at. These guys are all contacts, and you can talk to them about your program and whether you think they can give you information about their specific interests as they've written in the FOA, as well as information about how your project fits into the overall portfolio of pain within their institutes. But the important thing is we do take pretty much all pain indication areas for this program. Key resources that are available. I talked about the contracts that are available for the small molecules, but there's also, NCAT's intramural resources you could tap into. And I mentioned that a second ago, but the key thing there is you need to reach out and establish a CRADA with NCATs prior to submission of the application. They have to agree they want to enter into this research and development agreement with you and before you move forward. So you want to reach out to NCATs early, 6 to 8 weeks at least before the receipt date, to think about getting that established and figuring out what resources you're going to utilize from them if you're going to do that. Additional IC resources, different ICs on that list for may have other resources that they can help you with, and you talk to the science officers, and if they have them, you can include them if they agree to provide them. It is a phased mechanism. As I said, all eligible organizations are welcome to apply to the UG3, UH3, but everyone has to come to the UG3 first. So depending on where you're coming in along the spectrum from exploratory, hit to lead, lead optimization, IND enabling, will really dictate how long the grant will end up being total, but you have to enter the UG3 phase. And there's always stuff that needs to be taken care of in order to be ready for even the development project. There are things like dose range finding tox, getting the transporter panels and things like that are required for IND. All that stuff has to be taken care of, and so it can be part of the UG3 phase, part of the go, no go decision point to move forward to the UH3 phase. We also expect that, there'll be that component of the biomarker section that we talked about that will require additional effort to do, and it can be part of the UG3 and UH3 mechanism here. It is important to discuss your budget with NIH staff to make sure that you're accounting for things properly. Things that you're going to utilize our contracts are not part of your budget. But we need to make sure that what you're asking us to do, we can in fact do for you. So it's important to talk to us about your budget before submission, and make sure that if you have any questions that we can answer those appropriately. I do want to point this out. There's a notice I put out in November because there's some language that slipped into this RFA by accident about matching funds. There is no cost matching requirement for this RFA, and with effect of the March 24th date, the language that was erroneously put in the FOA has been rescinded, and there you by this notice. Activities that are not responsive. Screening to identify hit compounds, and those, that'll be part of the earlier FOAs when they are available. Basic research studies of disease mechanisms, animal model development, development of diagnostics and diagnostic devices, for which all of these presently have existing mechanisms and future funding mechanisms may be available later this year that are more HEAL specific, but NINDS has funding opportunities for all of these first 4 bullets. Studies directed beyond phase 1 clinical testing, and this is really important. So if you're coming in and saying, I'm going to do an efficacy study in the clinic, that's not going to be responsive to this application. And chances are you'd have to have obviously a properly funded clinical trial for that. And in most cases, just don't confuse an efficacy study with a biomarker read that you plan on doing. Opioid sparing projects and projects targeting mu opioid are not responsive to this application. We can talk about what that means, but anything essentially targeting mu opioid out of not responsive, we won't accept those applications. As I mentioned, 2-phase mechanism, we expect to start up to 17 projects. How do we go from phase, the UG3 phase to the UH3 phase? That's not done through another re-review through our peer review system. It's done through an internal administrative review. And it's going to be, as we said, we expect to advance up to 5 projects in that first few years to the UH3 phase. And that'll be based on the progress against the milestones, availability of funds, all checking all the boxes in terms of what we're looking for from a program. Do we have the biomarker? Do we have the target? Is it novel? All of that things will be part of the discussion as we determine from the portfolio which ones are most likely to move forward. The UG3 phase can be 1 to 2 years. All projects must enter that. Look carefully at the FOA. It explains, if you're coming in a discovery project, what's permissible. If you're coming in a development project, what's permissible in the UG3 phase. The goal in these cases is, to hopefully get a well-characterized lead candidate, either at the, in the medchem stage or in the development stage. But bottom line is, in the early stage program, we're trying to get to in vivo efficacy testing at the end, at a minimum, at the end of the UG3. The UH3 is any further optimization activities. The biomarker development studies will go on in earnest at this point. Non-GLP studies, tox studies, PK, formulation studies, cell bank development testing, gene level expression, biodistribution, tumorigenicity, immunogenicity, process development, manufacturing, clinical candidate for the IND and IND enabling safety, pharmacology, genotox, hERG, and tox studies are all things that's going to happen in that UH3 phase. So you can't come in and say, I'm going to immediately do IND enabling studies in this program, because it's going to be very difficult to execute that in year 1. So, and plus there's usually things that have to convince us in a milestone base that you're going to do that. We talked about the administrative review and all the different priorities there. So lastly, I want to touch on a few things you should do. You should read the funding opportunity very carefully. It has things that you must do, and it has things you should do. Things you must do, you must include budget seats for all years of the grants, both phases, UG3 and UH3, by year. So don't give me a budget that's just UG3 for 2 years and a budget for just UH3 for 3 years. And so you have 2 budgets. I need if it's 5 years, I need 5 sets of budget seats. And I can tell you in the last set of applications, half of them came in with budgets I had to go back to them and ask them to fix. And sometimes there's not any time left to do that. Clearly indicate what will be done as part of the grant and what will be expected from NIH contractors. It tells you in the forward to put this in a table and ask in this request for what should be in that table. Please follow those requests. Address the biomarker component. In our first round of applications, I would say a number of people didn't discuss the biomarker component. I don't know how that will do in review, but that's an important aspect of our program here, and we do want you to have a biomarker component. It should be addressing, is it feasible? If it's not feasible, why is it not feasible? If it is feasible, what are you going to do? Stick to the page limit, put forth a solid scientific premise, and address the rigor and reproducibility of your preliminary data and the data you're going to generate in the research strategy section. Address any obvious criticisms of the starting point. I suggest you contact your colleagues, NIH staff, if you're having trouble figuring out what those are, but anything that you think that someone might question, you should be upfront and saying, yes, we recognize this is a potential issue and this is how we think we're going to address that issue. Because silence on it, the assumption is you're not aware of the issue, and it tends to get dinged more heavily in review. I mentioned this already. I can't mention it enough. Do address the rigor and reproducibility of your preliminary and the data you plan on generating as part of your grant in the research strategy section. Don't bury it in the vertebrate section of your grant application. Complete all your registrations at least 6 to 8 weeks in advance. Consider submitting your application early. And I can tell you a number of people did this in the last round. And I went back to them and said, you have this error, that error in your application, like the budget sheets I was just mentioning. So you need to resubmit a complete set of budget sheets. And they had time to fix that before the deadline. Discuss your budget with us, NIH staff, if you have any questions at all. Talk to your business development folk and your tech transfer groups at your academic institutions, because you need to plan for funding of the grant, the patents, as well as any license activities down the road. That's really important to know, that we want to know what those plans are so that you are thinking way ahead, because we want you to eventually license this program, get money to take it further, so it doesn't end up on the shelf somewhere if you're successful. Some things not to do: don't plan on this to be your sole source of funding for your lab, because it's milestone driven and can end abruptly if you run into a scientific wall. Don't forget to address the NIH contract resources. Don't plan on contracts for disease biology, as I mentioned. Don't include contracts in your application budgets. Don't reach out to NIH contractors for letters of support, because I can't tell you which contractor you're going to have at the time we actually execute your particular contract. So putting a letter of support from our contractors does not help. So today I'm happy to answer questions about the application process, the review process, budgets and proposals, contracts, intellectual property, and we'll go there now. So Tasha, I'm gonna ask you to sort of put up the questions on your side for me, and I will start to answer those questions as best I can. And Michael will pop in whenever he can. Anne Marie's on the phone now as well, so we will try to answer as many of these questions as we go through.
So Chuck, the first question I see is from Yuri Miller. He says, how receptive would the program be to a biologic delivered intrathecally or versus systemically? Do you want to address it, or do you want me to address that?
Sorry, I've got Natasha also putting things up simultaneously. Sorry. So asking about intrathecal versus systemic. I mean, I think the key item there is, are you going to be able to deliver this in a way that is not a combination therapy out of the gate to phase 1? So if it's a combination device and therapeutic, that's not going to be in scope of what we're doing. But if it's going to just be IT administration via in the phase 1, then yes, it could be within there. But I would say you may want to consider the clinical path for that, and our contractors may not be the best choice. Because you may have more than minimal risk associated with that. So you just need to think through that carefully, and we can have a discussion about it.
The only thing I would add on to Chuck's answer is that the reviewers are going to address whether or not this compound or this pathway for treatment will have a significant impact on the patient population you're identifying. So if you're identifying a patient population that has access to, or it would be reasonable to use intrathecal administration, or that your treatment would last weeks to months, then that would be more reasonable than, let's say, weekly injections that were intrathecal. So these you should address in the application itself, because the reviewers are going to bring it up. But there's no part of the program itself that excludes intrathecal or systemic administration.
Correct. And, as Michael alludes to, you need to have your target product profile as part of the application. And, we can't stress enough that it's important to include clinicians in that development of that target product profile. So Tasha, can you slide back up to whatever my next question is? So if you'd like to schedule a one-on-one discussion, if you're small molecule space, contact me. If you're biologic space, contact Anne Marie. And those email addresses I'll put up in just a minute. In fact, I can put them up now if I can go down the page here. And we can certainly answer those for you and set up meetings. I'm presently without operations coordinator. It normally does my email stuff, but we are working on remedying that, and Tasha right now is helping me on the interim. So have some patience as we set up those calls. Are neuromodulation techniques allowed to study under HEAL program? So those would be devices. Yeah, go for it.
Let me address that. So NIH has many programs in the Brain Initiative, in the HEAL Initiative, as well as inside NINDS, where neuromodulation devices are responsive for that. This particular funding announcement is for small molecules and biologics. So feel free to reach out to me. My email is on the screen there, and then we'll hook you up with the right people and the appropriate funding announcement for neuromodulation devices.
Okay. For a proposal including GMP manufacturing, IND enabling studies, phase 1 clinical trial, which is all of them, what specific types of work should utilize the NIH contract resources rather than separate CROs? I would say it really depends. If you have something really specialized, then we would be open to having an outside CRO do that work. If it's something that's sort of a generic small molecule synthesis, and we could certainly do most of that stuff within our contract resources, which is the preferred method, but not the required method. So again, talking to us is important to figure out what it is about your program and why you'd want to use your own CROs versus the NIH CROs. And some of the biologics, those contracts are not yet established, and so you may have to plan for that. Can the project come directly to development stage, i.e. IND enabling non-clinical studies, GMP manufacturing, job cycles? So if you're coming directly to the development stage, chances are you really are beyond our program in many ways. You need to talk to me a bit more about what your package is, because we have limited budget in the UG3 phase, and we're not looking to just take something directly in to those clinical studies. And so my obvious question is, do you have a biomarker there? Do you have all the other components of the program that we expect to see there? So it's not just coming to say, I want to do IND enabling studies and go forward. So we need to talk about that a bit more.
I would add on to that there are other programs inside NINDS and the HEAL Initiative for funding those types of studies. So again, reach out to us, and this might not be the best funding announcement for those types of studies.
Exactly. I mean, we can potentially handle it within the program, but we really need to talk. And I found few people actually have everything they need for IND coming in, but we'll see. does a French company in collaboration with a US academic lab where the preclinical experiments will be done can apply this, and who will be the PI? Well, you would, all right, so that's unpack that. So yes, foreign institutions are eligible, as well as foreign components. Depending on who is the foreign institution would sort of dictate who the, whether this is a foreign application having to go through a second level of review by council or not. Who the PI is I think really up to the team to figure out based on what the program is exactly. But it could be either one. It could be the, it could be the U.S. institution. It could be the foreign institution, depending on how the grant is structured.
So the assessment of the foreign component that is done at council isn't a re-review of the application for its scientific merit. It's a review for the appropriateness for doing the project at a foreign site. And in my experience, it's usually better to have the PI be the domestic and then the foreign site be the secondary, but it's really dependent on the details, and I highly recommend that you reach out to us before the application. And in all these cases where Chuck and myself or Anne-Marie should be reached out, we're really talking about you reaching out to us 6 weeks or more before the application's due. Sometimes these require changes to the application, and it takes time to implement them.
Yep. Okay. Are there classes of molecules for which the funding opportunity is not available? For example, opioid compounds that are not classic mu agonists. If it's targeting the mu opioid receptor, it is not going to be responsive to our application or to our funding opportunity. If it is designed to be opioid sparing, it's not going to be responsive to it. But if you're targeting capi-opioid, yeah, no problem.
But the goal of this FOA is to really develop non-opioid treatments for pain. That is the goal of the program, and that's why these are not responsive. You can apply to other ways inside NIH for those programs or for those types of compounds. But the goal here is to really change the paradigm of how we treat pain.
Can you give examples of appropriate biomarkers? I guess that's an interesting thing. So if you were, you were targeting a specific receptor, and you had a way to tell in vivo that you've engaged that receptor, that kind of biomarker would be what we're looking for. I can think of the examples of things like TNF alpha and inflammation, right? Where you're not looking at a direct read of inflammation, but you're looking at TNF levels, something of that nature. We had a program, for instance, for our in neuroscience recently that was directed at our retinol binding protein 4 reduction, and they were able to monitor that in vivo and even in phase 1 and to see a reduction of that protein. So they knew they hit the target and had the desired effect. How receptive would the program be to a biologic delivered intrathecally or systemically? We already answered that question. Would short-term indications, perioperative pain, be seen as less significant than a long-term pain condition, neuropathic pain, or
I can address that. Yeah, so there is those acute pain, chronic pain, inflammatory pain, pain due to neuropathic pain, all of these are equally responsive to this funding announcement. There isn't an emphasis on one type of pain or another, because all of these types of pain conditions are contributing to the opioid epidemic. Okay, so the opioids during the perioperative period lead to then long-term opioid prescriptions. So the acute pain associated with surgery is a responsive application to the FOA.
Yes, indeed. Okay. Is Q, this Q is, okay, by IP. Does the PI have to be the patent holder. The PI institution has to have license to develop the program commercially. So no, it's not acceptable to say I want to develop Pfizer's compound for a new indication and then hope that Pfizer is going to give you a license at the end of the day, because they're more likely to give you a cease and desist order than a license in that scenario. So we have no idea whether we don't want to invest a whole lot of money in something that we don't know whether it can actually go anywhere. So we do want to know that you have a plan and a path to be able to commercialize the program at the end of the day. Can development of biomarker or can be studied aimed for, I think that the question is, can it be both part of the UG3 and the UH3 phase? And yes, I would expect it to be part of both phases. I expect probably the biggest cost to probably be in the UH3 phase as it is in a phased mechanism. Does the NIH have any contractors for virus production for gene therapy-based approaches for chronic pain? And Ray, you want to comment on sort of our ongoing efforts in this thing? Presently, we do not, but we have plans.
Yeah, we are actually in the process of establishing contracts with both oligo-based and viral-based gene therapy CROs. They should be online within this next year for that, and you can certainly contact to discuss some of the activities that can be done during this manufacturing.
So by the time you get to the UH3 phase, we anticipate that those will be there.
In place.
Can we apply if we already have a grant working with NCATS to look at preclinical compound? I'm not sure exactly what that question.
I think I can address that. I think I got it. So there's a collaboration with an intramural group at NCATS to, you didn't give the details of what they're doing, but there's an intramural group. So in a collaboration between intramural and extramural is responsive to this funding announcement. As long as the activities that you'll be doing in this funding announcement don't overlap with the activities you're doing with NCATS currently, then it's a responsive application. And we'll just now, if there's going to be NCATS you're planning on collaborating with them as a part of this work for ongoing studies in it, that's going to have to be a discussion you do on the front end, and then we have to do some budgeting gymnastics on our end and make sure that you write the application appropriately. So let's just say we have to discuss it, and I hope that gives you a clear answer.
I think it's something we could certainly work through because NCATS is part of this program and is interested in setting up CRADAs and collaborating on this program. And so if they already have an established agreement with you, then having the discussion about how you could leverage this program with them would be a good starting point. And then talking to us about that and how we could execute the application and the budgets appropriately would be the next step. Okay, I am a CEO of a small company, and I also take research. I also take a research science position at a university as a full-time employee. Can I apply to a grant as a PI to do ADMET study and ask my professor at NYU to be a subaward PI to do the efficacy studies? Yeah, probably. Again, the devil's in the details. Who's going to be the grantee award institution? Who's going to own the intellectual property that gets developed? Who's going to prosecute that property? That's the part that you would need to have all worked out. There's no preference here on how you make the organization or the agreement together. It's just as long as there is a clear path to taking the property forward. Remember, the purpose of the program at the end of the day is to identify a clinical candidate, take it into the clinic, and hopefully also develop an efficacy or target engagement biomarker. So at this point, I don't see any other questions from there. And if, I'm happy to answer any more questions if people have it. I'll give it a couple of minutes for people to type in a question. But if you had your questions answered already, then I will say this: we'll have a recording of this webinar hopefully available in the not too distant future. But I will guarantee you I will have a PDF of the slides I presented today available to you, and will be sent out to all the people who registered today at the email addresses.
There's one additional question I see here about biomarkers.
Yeah, our imaging markers,
Yep.
for example, this synovial joint arthritic fluid could be considered a biomarker. I would say, yeah, probably.
These are biomarkers, but they're not pharmacodynamic biomarkers unless
Well,
it's
it could
in
be
direct response to the mechanism of your therapeutic. If it's something very far downstream, then that's not the type of marker we're looking for here. We're not looking for quote unquote general biomarkers for pain. We're looking for pharmacodynamic markers of the action of the small molecule or the biologic themselves.
Right, because I can think of indications that you would do, for instance, where you inject, potentially a long-term therapy in the knee, and you may actually do imaging in the knee to see the pharmacodynamic effects of that. But that's sort of, again, that wasn't the detail of exactly how you do it, but yes, it's theoretically possible that that could be within scope. Well, if I don't have any other questions, I want to again thank you all for taking the time to learn more about this program. Our next receipt date's in March, and then many more to come through June of 2022. So I do hope that you consider our program. If you have any questions, you reach out to, Anne-Marie, Michael, or myself, and we can tell you more about the program and hopefully answer your questions going forward on it. Nothing else has come in, so I guess we'll end the seminar now. Thank you very much all for your attention today. Bye now. Thank you, Michael. Thank you, Anne-Marie.